Hi, my name is Chloe Ghent. I am a third year graduate student at UCSF. I'm originally from Washington State and I did my undergrad at UC Berkeley. I'm currently doing my thesis work in Denitsa Fujimori's lab, and our lab is generally interested in characterizing post-transcriptional and post-translational modifications of both chromatin and the bacterial ribosome. And I'm interested in specifically how bacterial ribosome modifications operate in the context of antibiotic resistance. There's a continued and ongoing rise in antibiotic resistance worldwide across many bacterial strains, and it's predicted that deaths attributable to this resistance will also continue to increase. And so this poses a pretty substantial public health threat and necessitates a better understanding of antibiotic resistance mechanisms in order to engineer future antibiotic classes that can circumvent these pathways. So a pretty typical mechanism of action for antibiotics is to bind directly to the bacterial ribosome and cause it to stall along mRNA transcripts and inhibit protein translation. Subsequently, many antibiotic resistance genes encode proteins that can modify the ribosome and occlude antibiotic binding to allow for protein synthesis to continue. The specific resistance gene I'm interested in is called CFR, and it confers resistance to many classes of antibiotics. And there's evidence in the literature that resistance conferred by CFR can be induced, suggesting that the expression and activity of CFR can be sensitized to the presence of antibiotics. So CFR activity can reach its maximum capacity when it is most needed. And there's some unique sequence features upstream of the CFR gene that occur in many natural contexts that hint at mechanisms through which this regulation can occur. And my thesis project essentially is focused on better understanding how CFR expression and activity is regulated. There are two models of regulation through which I hypothesize CFR activity and expression is modulated. In the first model, the presence of antibiotic is necessary to allow the ribosome access to the CFR transcript and subsequently regulates CFR at the protein's translation level. In my second model, the presence of antibiotic is necessary to regulate the expression of a CFR inhibitor and subsequently regulates CFR at the protein activity level. And so I am planning to use a combination of both in vitro biochemical and microbiology techniques to test my two hypothesized models and hopefully better characterize the regulation of CFR and also provide more insight more generally into antibiotic resistance mechanisms.